Tonight's lecture is over Chapter 3, Important Biological Macromolecules. A cell is mostly water, but the rest of it consists mainly of carbon-based molecules. So why are carbon atoms so versatile as molecular ingredients? Recall that an atom's bonding ability is related to the number of electrons it must share to complete its outer shell. A carbon atom has 4 electrons in an outer shell that holds 8. Carbon completes its outer shell by sharing electrons with other atoms in 4 covalent bonds. Thus, each carbon atom acts as an intersection from which an organic compound can branch off up to 4 different directions. And because carbon can use one or more of its bonds to attach to other carbon atoms, it is possible to construct an endless diversity of carbon skeletons varying in size and branching patterns. The carbon atoms of organic compounds can also use one or more of their bonds to partner with other elements, most commonly hydrogen, oxygen, and nitrogen. In terms of chemical composition, the simplest organic compounds are hydrocarbons, which contain only carbon and hydrogen atoms. And the simplest hydrocarbon is methane. We see methane in the upper right-hand corner of this slide. This is a single carbon atom bonded to 4 hydrogen atoms. Methane is one of the most abundant hydrocarbons in natural gas and is also produced by prokaryotes that live in swamps and the digestive tracts of grazing animals such as cows. Larger hydrocarbons, such as octane with 8 carbons, are the main molecules in the gasoline we burn in cars and other machines. Hydrocarbons are also important fuels in your body. The energy-rich parts of fat molecules have hydrocarbon structures. Each type of organic compound has a unique 3-dimensional shape. So in the lower right-hand corner of this slide, we see various molecular structures for carbon molecules. Moving on to the structure and function of carbohydrates. Carbohydrates, commonly known as carbs, are sugars or sugar polymers. Some examples are small sugar molecules dissolved in soft drinks and large starch molecules in pasta and potatoes. In animals, carbohydrates are a primary source of dietary energy. In plants, they serve as a building material for much of the plant body. Carbohydrates always appear in a carbon to hydrogen to oxygen ratio that is 1 to 2 to 1. For instance, the molecular formula for glucose is C6H12O6. That means 6 carbons, 12 hydrogens, and 6 oxygens. And as detailed on this slide, simple sugars such as monosaccharides can be dehydrated and create longer chains of sugars such as disaccharides and polysaccharides. So by removing one molecule of water, we can link together 2 monosaccharides and form a disaccharide. Dehydration reactions can be reversed via a process called hydrolysis. An example of a hydrolysis reaction is the breakdown of lactose by the enzyme lactase. Lactase promotes the hydrolysis of lactose into monomers. Moving on to monosaccharides. Monosaccharides, or simple sugars, cannot be broken down by hydrolysis into smaller sugars. Common examples are glucose and fructose. Glucose can be found in sports drinks and fructose can be found in fruit. Both of these simple sugars are also in honey. The molecular formula of glucose is C6H12O6, and fructose has the same formula, but its atoms are arranged differently. Glucose and fructose are examples of isomers, that is, molecules that have the same molecular formula but different structures. The shape of a molecule is very important, and seemingly minor differences in the arrangement of atoms give isomers different properties. In this case, the rearrangement of the functional groups makes fructose taste considerably sweeter than glucose. Monosaccharides, particularly glucose, are the main fuel molecules for cellular work. Analogous to an automobile engine consuming gasoline, your cells break down glucose molecules and extract their stored energy, giving off carbon dioxide as exhaust. The rapid conversion of glucose to cellular energy is why an aqueous solution of glucose, often called dextrose, is injected into the bloodstream of sick or injured patients. The glucose provides an immediate energy source to tissues in need of repair. In addition to their use as an energy source, monosaccharides provide cells with carbon skeletons that can be used as a raw material for manufacturing other kinds of organic compounds. A disaccharide, or double sugar, is constructed from two monosaccharides through a dehydration reaction. Lactose, or milk sugar, is made from the monosaccharides glucose and galactose. Another common disaccharide is maltose. This is naturally found in germinating seeds. Maltose is used in making beer, malt whiskey, liquor, malted milkshakes, and malted milk ball candy. A molecule of maltose consists of two glucose monomers joined together. However, the most common disaccharide is sucrose. This is common table sugar. This consists of a glucose linked to a fructose. Sucrose is the main carbohydrate in plant sap, and it nourishes all parts of the plant. Sucrose is extracted from the stems of sugarcane or the roots of sugar beets. However, sucrose is rarely used as a sweetener in processed foods in the U.S. Much more common is high fructose corn syrup, made through a commercial process that converts natural glucose in corn syrup to much sweeter fructose. If you read the label on a soft drink can or bottle, you'll likely find that high fructose corn syrup is one of the first ingredients listed. The United States is one of the world's leading markets for sweeteners, with the average American consuming 45 kilograms, that's 100 pounds, per year. And this is consumed mainly as sucrose and high fructose corn syrup. The national quote-unquote sweet tooth persists in spite of our growing awareness about how sugar can negatively affect our health. Sugar is a major cause of tooth decay. Moreover, high sugar consumption tends to replace eating more varied and nutritious foods. The description of sugars as quote-unquote empty calories is accurate in the sense that most sweeteners contain only negligible amount of nutrients other than carbohydrates. For good health, we also require proteins, fats, vitamins, and minerals. Plus, we need to include substantial amounts of complex carbohydrates, that is, polysaccharides. With that being said, we'll now discuss polysaccharides. Complex carbohydrates, or polysaccharides, are long chains of sugar units. These are polymers of monosaccharides. One familiar example of a polysaccharide is starch. Starch is found in plants. Starch consists of many glucose monomers strung together. Plant cells store starch in granules where it is available as a sugar stockpile that can be broken down as needed to provide energy and raw material for building other molecules. For instance, potatoes and grains, such as wheat, corn, and rice, are the major sources of starch in the human diet. Humans and most other animals can use starch as food because their digestive systems break down the bonds between glucose monomers through hydrolysis. Animals store excess sugar in the form of a polysaccharide called glycogen. Glycogen and starch are similar in structure. They are both polymers of glucose monomers, but glycogen is more extensively branched. Most of our glycogen is stored as granules in our liver and muscle cells, which break down the glycogen to release glucose when it is needed for energy. This is the basis for carbo loading, the consumption of large amounts of starchy foods the night before an athletic event. The starch is converted to glycogen, which is then available for rapid use during physical activity the next day. In addition to playing an important role in nutrition, certain polysaccharides serve as structural components. For instance, cellulose is the most abundant organic compound on Earth. It forms cable-like fibrils in the tough walls that enclose plant cells and is a major component of wood. We take advantage of that structural strength when we use lumber as a building material. Cellulose resembles starch and glycogen in being a polymer of glucose, but its glucose monomers are linked together in a different orientation. Unlike the glucose linkages in starch and glycogen, those in cellulose cannot be broken down by most animals. The cellulose in plant foods, which passes unchanged through our digestive tract, is commonly known as dietary fiber. Because it remains undigested, fiber does not serve as a nutrient, but it does appear to help keep our digestive system healthy. Most Americans do not get the recommended levels of fiber in their diet. Foods rich in fiber include fruits and vegetables, whole grain, bran, and beans. Grazing animals and wood-eating insects such as termites can derive nutrition from cellulose. They have prokaryotes inhabiting their digestive tracts that help to break down the cellulose. Monosaccharides such as glucose and fructose and disaccharides such as sucrose and lactose dissolve readily in water. This forms a sugary solution such as you would find in a soft drink. In contrast, cellulose does not dissolve in water. In spite of this difference, almost all carbohydrates are hydrophilic. The hydrophilic nature of cellulose makes a bath towel fluffy and water absorbent. Moving on to lipids, in contrast to carbohydrates and most other biological molecules, lipids are organic compounds that are hydrophobic. In other words, they do not mix well with water. You have probably observed this chemical behavior in an unshaken bottle of salad dressing. The oil, which is a type of lipid, separates from the vinegar, which is mostly water. If you shake the bottle, you can force a temporary mixture long enough to douse your salad with the dressing, but what remains in the bottle will quickly separate again when you stop shaking it. A typical fat consists of a glycerol molecule joined with 3 fatty acid molecules via dehydration reactions. The resulting fat is called a triglyceride, a term you may see in the results of medical tests for fat in the blood. The major portion of a fatty acid is a long hydrocarbon that stores quite a bit of energy, like the hydrocarbons of gasoline. In fact, a pound of fat packs more than twice as much energy as a pound of carbohydrate. The downside to this energy efficiency is that it is very difficult for the person trying to lose weight to quote-unquote burn off excess fat. It is important to understand that a reasonable amount of body fat is both normal and healthy as a fuel reserve. We stock these long-term food stores in specialized reservoirs called adipose cells, which swell and shrink when we deposit and withdraw fat from them. The adipose tissue, or body fat, not only stores energy but also cushions vital organs and insulates us. This helps us to maintain warm body temperatures even when the outside air is cold. Moving on to the next slide. Notice in the bottom figure the unsaturated fatty acid bends where there is a double bond in the hydrocarbon skeleton. The fatty acid is said to be unsaturated because it has fewer than the maximum numbers of hydrogens at the location of the double bond. The other 2 fatty acids in the fat molecule lack double bonds in their hydrocarbon portions. These fatty acids are saturated, meaning they contain the maximum number of hydrogen atoms. And you can see a drawing of the saturated fatty acid on the left-hand side of this slide. So a saturated fat is one with all 3 of its fatty acid tails saturated. If one or more of the fatty acids is unsaturated, then it is an unsaturated fat. Moving on to the next slide, most animal fats, such as lard and butter, have a relatively high proportion of saturated fatty acids. The linear shape of saturated fatty acids allows these molecules to stack easily, so saturated fats tend to be solid at room temperature. Diets rich in saturated fats may contribute to cardiovascular disease by promoting arteriosclerosis. In this condition, lipid-containing deposits called plaque build up along the inside of the walls of blood vessels, and this reduces blood flow, and it thereby increases the risk of heart attacks and strokes. In contrast to red meat and dairy products, plant and fish fats are relatively high in unsaturated fatty acids. The bent shape of unsaturated fatty acids makes them less likely to form solids, so most unsaturated fats are liquid at room temperature. Examples of oils that are primarily unsaturated include vegetable oils and fish oils. Although plant oils tend to be low in saturated fat, tropical plant fats are an exception. Cocoa butter, a main ingredient in chocolate, contains a mix of saturated and unsaturated fat that gives it a melting point near body temperature. Thus, chocolate stays solid at room temperature, but then melts in your mouth. Moving on to phospholipids. Phospholipids are the major constituent of the plasma membrane. Like fats, they are composed of fatty acid chains linked to a glycerol or some similar backbone. Instead of 3 fatty acids attached, there are only 2 fatty acids, and a third carbon of the glycerol backbone is bound to a phosphate group. The phosphate group is modified by the addition of an alcohol. A phospholipid has both hydrophobic and hydrophilic regions. The fatty acid chains are hydrophobic and exclude themselves from water, whereas the phosphate is hydrophilic and interacts with water. Cells are surrounded by a membrane which has a bilayer of phospholipids. The fatty acids of phospholipids face inside, away from the water, whereas the phosphate group can face either the outside environment or the inside of the cell, which are both aqueous. Moving on to steroids and waxes. Steroids are classified as lipids because they are hydrophobic. However, steroids are very different from fats in structure and function. All steroids have a carbon skeleton that is bent to form 4 fused rings. Different steroids vary in the functional groups attached to the core set of rings, and these variations affect their function. One common steroid is cholesterol, which has a bad reputation because of its association with cardiovascular disease. However, cholesterol is an essential molecule in your body. It is a key component of the membranes that surround your cells. It is also the base steroid from which your body produces other steroids such as the sex hormones estrogen and testosterone. On the other hand, waxes are very resistant to water, and we see 3 examples of waxes on our slide: beeswax, lanolin, and leaf cuticle. Moving on to proteins. Proteins are the most elaborate of life's molecules. A protein is a polymer constructed from amino acid monomers. Your body has tens of thousands of different kinds of proteins, each with a unique 3-dimensional shape corresponding to a specific function. All proteins are macromolecules constructed from a common set of 20 kinds of amino acids. Each amino acid consists of a central carbon atom bound to 4 covalent partners. 3 of those attachments are common to all 20 amino acids. That is the carboxyl group, the amino group, and a hydrogen atom. The variable component of amino acids is the side group, which is attached to the 4th bond of the central carbon. Each type of amino acid has a unique side group, giving that amino acid its special chemical properties. Proteins perform most of the tasks required for life. Perhaps their most important role is as enzymes. Enzymes are chemicals that change the rate of a chemical reaction without being changed in the process. Remember earlier during this lecture we were talking about the enzyme lactase. Lactase is just one of thousands of enzymes produced by the cells in your body. Chemically speaking, hormones may be classified as either proteins or steroids. All of the hormones in the human body except the sex hormones and those from the adrenal cortex are proteins or protein derivatives. Antibodies are also another example of a protein, and antibodies are important to your immune system. Moving on to protein structure, cells link amino acid monomers together by dehydration reactions. The bond between adjacent amino acids is called a peptide bond. The resulting long chain of amino acids is called a polypeptide. A protein is a polymer consisting of one or more polypeptides. Your body has tens of thousands of different kinds of proteins. How is it possible to make such a huge variety of proteins from just 20 kinds of amino acids? The answer is arrangement. You know that you can make many different words by varying the sequence of just 26 letters in the English alphabet. So just as each word that is constructed from a unique succession of letters, each protein has a unique linear sequence of amino acids. This specific amino acid sequence is called the protein's primary structure. Every protein has a specific amino acid sequence. And this is very important. For instance, in the English language, changing one letter can drastically affect the meaning of a word. For instance, the word tasty. If we change the first T in tasty to an N, we get nasty. Those two words have completely different meanings. And it's similar with proteins. Even a slight change in the primary structure can affect a protein's ability to function. For example, the substitution of one amino acid for another at a particular position in hemoglobin, the blood protein that carries oxygen, causes sickle cell disease. This is an inherited blood disorder. So certain stretches of the polypeptide form local patterns called a secondary structure. Two types are named, the alpha helix and the pleated sheet. The secondary structure of protein is reinforced by hydrogen bonds. These hydrogen bonds occur along the polypeptide backbone. The overall three-dimensional shape of the polypeptide is called a tertiary structure. It is reinforced by chemical bonds between the side groups of amino acids in the different regions of the polypeptide chain. And the quaternary structure of proteins has two or more polypeptide chains. This results from bonds between the chains. Moving on to the next slide, a protein's shape is sensitive to the surrounding environment. An unfavorable change in temperature, pH, or some other quality of the environment can cause a protein to unravel and lose its normal shape. This is called denaturation of a protein. If you cook an egg, the transformation of the egg white from clear to opaque is caused by the proteins in the egg white denaturing. The denatured proteins become insoluble in water and form a white solid. One of the reasons why extremely high fevers are so dangerous is that some proteins in the body become denatured above about 104 degrees Fahrenheit. And this next slide has a table that illustrates some of the functions of proteins. So we see we've got three columns. We've got the type of protein, an example of the protein, and then the function of the protein. So for instance, digestive enzymes, some examples of a digestive enzyme would be amylase, lipase, pepsin, trypsin, and they help to digest food in the body by catabolizing nutrients into units. So take a few minutes and go through this table on your own time. Nucleic acids are macromolecules that provide the directions for building proteins. The name nucleic comes from their location in the nuclei of eukaryotic cells. There are actually two types of nucleic acids: DNA, which stands for deoxyribose nucleic acid, and RNA, which stands for ribonucleic acid. The genetic material that humans and other organisms inherit from their parents consists of giant molecules of DNA. The DNA resides in the cell as one or more very long fibers called chromosomes. A gene is a specific stretch of DNA that programs the amino acid sequence of a polypeptide. Those programmed instructions, however, are written in a kind of chemical code that must be translated from quote-unquote nucleic acid language to quote-unquote protein language. Nucleic acids are polymers made from monomers called nucleotides. Each nucleotide contains 3 parts. At the center of each nucleotide is a 5-carbon sugar, deoxyribose in DNA and ribose in RNA. Attached to the sugar is a negatively charged phosphate group containing a phosphorus atom bonded to oxygen atoms. Also attached to the sugar is a nitrogenous base made of 1 or 2 rings. It is called a base because it accepts hydrogen ions in an aqueous solution. The sugar and phosphate are the same in all nucleotides, only the base varies. Each DNA nucleotide has one of 4 nitrogenous bases: adenine, abbreviated A, guanine, abbreviated G, cytosine, abbreviated C, and thymine, abbreviated T. Thus, all genetic information is written in a 4-letter alphabet. So how does RNA differ from DNA? Well, as its name implies, ribonucleic acid is a sugar called ribose rather than deoxyribose. Another difference between RNA and DNA is that instead of the base thymine, RNA has a similar but distinct base called uracil. Except for the presence of ribose and uracil, an RNA polynucleotide chain is identical to a DNA polynucleotide chain. However, RNA is usually found in the single-strand form, whereas DNA usually exists as a double helix. So after tonight's lecture, you should be able to answer these following questions. Why is it said that life is carbon-based? What kind of bonds does carbon form with other atoms? How can you distinguish between monosaccharides, disaccharides, and polysaccharides? What functions do carbohydrates have? Identify the different types of lipids that we discussed, their functions, and the health aspects of each. What monomers make up proteins? What are the 4 protein structures? You should be able to identify several major functions of different proteins. You should know what makes up a monomer of nucleic acids. You should also know the differences and similarities between the structure of DNA and RNA. And furthermore, you should know the function of DNA and RNA.